Hi, everyone. So it seems we momentarily lost Siobhan here. I'm just quickly going to take a moment to get her back online. Here we go.
Hi. Can everyone hear me? If I can just get some thumbs up in the comments. Great. Okay. So welcome everyone to our special edition tonight. We thought we should give a dual mention to the dark force out there that's currently clouding the entire world. So we're really glad to have you all join us this evening, and we're really looking forward to Pedro's talk. So for those of you who don't know us, my name is Siobhan, and Julianne is will also be on screen. She'll be introducing our speaker soon. And you can definitely look out for Darby in the comments. The Neural Engineering Research Venture is an online platform, and we aim to bring neuroscience to Africa and the world. And this is our way of doing it. Before we get going, we have some exciting announcements. Kaggle has a really cool competition on neuroimaging. You should definitely check that out. The prize money is great. Also, in case you're running out of reading during lockdown, Springer has released over 60 machine learning books. They also cover data science and everything. So definitely check those out. And then last, but certainly not least, if you ever find yourself in Cape Town, there is a great Cape Town meetup, the Cape Town Intelligence Systems, and their focus is on AI, and just something to look forward to in a world that has less social distancing. Before I hand over to Julianne, just some house rules. Please remember that we all come from very different backgrounds, so let's remember inclusive and respectful language. We welcome all questions. So there is an ask a question feature. If you can just post your question there, and then remember to up and down vote on your favorite questions. Oh no, you can't downvote, sorry, just upvote. So just remember to get that going. The most popular questions will be asked first. And if you are not able to come on screen, that's not a problem. Please just add please ask so that we know that to ask the question for you. If you're struggling with your connection, at risk of quoting the IT crowd, turning it off and on again really does work. So you can just refresh your browser and then it should work. And if that really doesn't...
Okay, it seems like we lost Siobhan again, ironically on the connection slide. So I'm going to jump straight into introducing our speaker for the evening. We are very excited to have Pedro Herrero-Vidal with us tonight. He is currently a neuroscience PhD student at New York University, and he has a background in biology, physics, and neuroscience with specific applications in the understanding of brain function. His current research includes the encoding of sensory stimuli into neural activity to illustrate how experience shapes these representations. So without further ado, I'm now going to invite Pedro on screen.
Thank you for the lovely introduction. I'm going to try to share my screen now. Can you all see it? So let me figure out a second how to share these properly, and I'll get started. So now, can you all see my screen? Hope so. I'm Pedro, and I'm going to be talking to you about the decoding of chemical information from population of olfactory neurons. And before I jump into it, I want to give you a slightly further introduction into about me. I did my undergrad in biochemistry at the University Autonomous of Madrid, where I also received a master's in physics, during which time I studied brain function and dysfunction across several levels, Then I came here to New York to do a master's in bioinformatics. And I also studied neuroscience during this time, learning the mechanisms that underlie associative memory. And I'm currently pursuing my PhD at NYU, advised by Christina Sabin and Dima Ringberg, studying encoding and decoding of other objects across the olfactory system. We're here to talk about the brain, but before that, I wanted to give you a brief perspective on how the fields of neuroscience and AI have helped each other. Importantly, here, when I talk about neuroscience, I refer to both experimental and theoretical neuroscience, and when I mention AI, I refer more broadly to the fields of statistics, computer science, and machine learning. Since the dawn of computers, neuroscience has served as inspiration for AI. And here we have an example of these interactions where neuroscience understood that neurons, here a schematic of them, integrate information, receive inputs from their dendrites, and then add them together in a weighted sum, after which they decide to trigger an action potential through a step function. Pretty much like it, they took inspiration from it and developed a perceptron algorithm where an putative neuron or artificial neuron takes weighted input sums of inputs, and after which we've passed it to an activation function to resolve complex, or in this case, very simple task, binary decision, giving an output. After, with the developing of other activation functions and backpropagation, we could stack several of these perceptrons and get to further and more complicated computations. Still, another example of these interactions that brought a revolution into the field of AI came once again from neuroscience, and this time took inspiration from the visual system. Here, and the neuroscience experiments that inspired these were carried by Hubel and Wiesel, that record their activity from the ventral visual stream of the cat while they were presenting stimuli, and they identified across this system several kinds of cells with different receptive fields, which led them to believe that they existed simple cells and complex cells, where simple cells had simple, for the redundancy, the receptive fields, in this case, in a particular area, the LGN centers around receptive fields that then were integrated by complex cells, forming more complicated, more integrated receptive fields. Some of these ideas, along with the idea of transitional invariance, serve of inspiration for Jan LeCun, who developed convolutional neural networks that use convolution, pooling, and sparse weighting hierarchies to perform complex computations. The number of examples that we can draw of these interactions are almost countless. Another one being the reward association, which inspire reinforcement learning, and so many more, as task generalization performed by animals, which inspire transfer learning. But today, I want to draw a little bit attention to the flow of information from AI to neuroscience. As an example of this interaction, I want to show you this beautiful experiment where, using these same convolutional neural networks that were inspired by the brain, now we can use them as models of the visual system. Here, what they did is they trained a neural network that mimicked the ventral visual stream, and while they were recording from macaques, they associated one of these units in this network to a particular neuron in the brain, and now they had a system to define an objective function and perform gradient descent to find the perfect tuning for these cells. Impressively, this feed-forward system does really well at capturing the activity of these neurons in the visual system. And here I'm showing you some of the examples of what these receptive fields for neurons in this area look like. But it's not only AI, the field that is suffering or experimenting a revolution. Neuroscience is also going through an incredible period of changing with the emergence of new recording techniques. These allow us to monitor simultaneously the activity of hundreds of neurons at a time or several brain regions in one snap. To name a few of these technologies, we can look at multi-unit recordings or calcium imaging or fMRI. And now we're faced with the task of understanding how those neurons are encoding information and performing these computations. To understand this better, now we're taking tools from AI and bringing them to neuroscience as a way to leverage this gap. And this is what I'm going to be focusing on today. To give you an outline of this talk, today I'm going to be talking to you first about the using of some of these machine learning methods to understand neural computation. In particular, I'll be talking about low-dimensional systems. And then I'm going to give you an example of this application in my own research, where we take those methods and use them for technical development. Moving on to the first topic. First, what does the brain do? The brain is in charge from taking a sensory stimulus for which it has to generate a meaningful neural representation to generate an adequate behavior. And as neuroscientists are tasked with understanding how does this process happen. And we do that in labs by training animals to perform complicated tasks while looking at neural activity. Here is a dummy example where each of these lines represent a spike over time, and each of the rows represents a neuron. So how do we go about understanding these patterns of activity and relating them to this bigger process? A way to think about it, as I say, is with low-dimensional methods or latent space models. And here's the generative model. We start from the bottom, where we have our observations, in this case, spike trains, which are high-dimensional, but importantly, they are measurable, and we can look at them, and they're also noisy. And one can think that these are generated from an internal process or computation that underlies it and is influenced by both the stimulus and the behavioral output, as well as the circuit in which it's embedded. And furthermore, for this to be a dimensionality reduction problem and increase interpretability, the dimensionality of this latent space or set of factors is lower dimensional than the observation space. Is this assumption reasonable? I'm going to argue yes, and you're going to see some examples during this talk, but briefly, there's experimental evidence showing that the activity of neurons is not totally independent, it's correlated, and therefore these correlations could explain why we can extract information from a subset of them. In most experimental conditions, the stimulus and behavior are usually also low-dimensional, which constrain the representation. And furthermore, simulations show that networks with a low-rank connectivity show low-dimensional dynamics as well, or low-dimensional representations, but are still powerful computational machines. To make this a little more concrete, what do these models look like? For the latent representation, we can start with something simple as a Gaussian distribution. And then what is important is how we generate observations from this model that could be, once again, really simple, described by this mapping function, a linear transformation of this latent space. However, as I said, neurons are noisy, and one can get sophisticated and model this noise as well. And depending on which specific decisions you make about the noise, you can end up in a model where you have no noise, PCA, which I assume you're all familiar with, but you can also, depending on how flexible you make it be, probabilistic PCA or factor analysis, where in probabilistic PCA, we assume that the noise of all neurons is the same. And in factor analysis, we allow for independent noise for individual neurons. This assumption is okay, but I said that the spike trains evolve over time, and so those stimulus and behaviors. So can we get a little more sophisticated when describing these latent dynamics? And indeed we can. Once again, in simple examples, we can define transitions over time for the latent trajectories as a linear function and added noise. When doing this, we end up with linear dynamical systems. One can get even more sophisticated when doing this, and you can use a Gaussian process prior for the latent dynamics, and ending with something like Gaussian process factor analysis, or you can even embed an entire recurrent neural network as the source of these dynamics, and as it was shown in this algorithm called LFATS. Now, I've tried to convince you that these models are useful and they could help us understand neural function, but can they actually do so? So to illustrate this, I'm going to bring you two examples. The first one relating to the encoding of sensory information, in particular, odor information in the locus. And to do so, they record the neural activity in the early olfactory system of these animals while presenting different odors. Here you see, as I said, that the responses of individual neurons, here each row, are really complicated and hard to understand. Here I'm showing you responses from this study where they present 3 different odors at a range of concentration where the presentation is aligned to this gray bar. So how can we drive insight from these recordings? In this article, they resource to one of these methods to generate a latent space, where I want to show you here that they found that different odors indicated by the 3 different colors span across different dimensions in this latent representation, which indicates that different populations are responsible for the encoding of different odors. These trajectories evolve over time, as so do the spike activity, and they start, it's hard to parse in this representation, but they start here evolving this semicircular trajectory and go back to its original state. Another observation that they made when looking at these responses is that as much as different odors evoke different population responses, there was something that was preserved in this neural computation when presenting the same odor at different concentrations, as you can see here by the different shades of color, or even more clearly here at the bottom, where larger concentrations span the same dimensions of this space, but the magnitude of these trajectories differs. Now, to bring you a second example, how they not only relate to the encoding of information, but also to decoding of how this is parsed at a behavioral output, I'm going to show you this other example. Here, they train animals, monkeys, to perform a task where they have to first poke the screen for a given amount of time, after which a target appears and it starts vibrating, signaling that the animal cannot move. And after the vibration stops, animals define a movement towards the target spot that appear on the screen. Here's a summary of this behavior, where, as you can see, the monkey performs this task really well, starting at the center where they're asked to perform the task and evolving to the different trajectories really precisely. So once again, we're interested in the neural computation, and to do so, they recorded from a motor region in the brain of these monkeys. And here I'm showing you three different neurons, three example neurons from this article. And as you can see, this area responds to movement, as well as it shows a preparatory movement. Here, movement is indicated by this second dot, and the appearance of the first target by the first dot. But is this all that we can extract from these representations? As you may guess, the answer is there's more. And as a first iteration from this data, they showed that actually the driving force for this neural processing was indeed an oscillatory trajectory that defines the movement. And this oscillatory component described in the populations is independent of the individual trajectory that the animal is performing at the level of the behavior. This gives us an intuition of this capacity, but more recently, more sophisticated methods using, as I showed you before, the recurrent neural networks help us to drive even further insight into these representations, when here again, a color schematic of the animal movement, so it's equivalent to this graph on the left, but this time on the right, here the latent structure predicted from these trajectories on a trial-by-trial basis, which indicate us that we can predict animal movement or the animal response for each given iteration of this procedure with fine detail. This is great, and here I've shown you two examples on how these kind of methods can drive us a further understanding of these representations by helping us visualize these processes. But are these just an after effect, or do they have a more conceptual meaning than just helping us represent neural data? To illustrate a more causal effect of these representations, I'm gonna keep on going with the motor examples. In this time, the motor output is not animal movement, but is a brain-computer interface. In these experiments, they use neural recordings from this same brain region that drive animal motion, and then perform dimensionality reduction to extract a meaningful intrinsic manifold of the representation. And just by feeding those inputs to a brain-computer interface, these animals were able to control the cursor on the screen and reach out to the particular targets showed on the screen. Here, in this other graph, is the particular trajectories described by the brain-computer interface guided by this neural activity, once again, simplified or represented through this latent representation. So there I show you that it's actually a great tool to help us understand causal effect of these neural representations. But beyond that, they also tell us about the computations of the circuit is performing. Hinting on this preparation, these authors claim that brains have billions of neurons. So, and when we're recording them, we only have access to a fraction of them. So if we, although with these generative models, we can extract relevant information, we don't have access to the entirety of the neurons. But in this brain-computer interface, because the motor output is driven only by the neurons that we're looking at, we can make further more causal statements about the network that is representing and driving this stimulus. To illustrate this process, they're recording from neurons, and then they're defining this latent representation, which if we start with 3 neurons for this example, this toy example, all the responses are occupy a single two-dimensional plane that has lower structure than the original space. So now what they did is they can modify the structure of this brain-computer interface control in a way that the perturbations or perturb the system such that the manipulations lay in this space, as so here in red, which is what is being shown that the neurons are constrained to, or outside of this particular latent structure. With this set of experiments, they were able to investigate network learning and rearrangement and the properties of the circuit. And here I'm showing you the main result, where at the beginning, here's the ability of the animal to perform the task, meaning starting from an original point and moving to the correct target signal on the screen. But after performing a perturbation of the control, where we change the accesses of used by this neural population still within this plane, the animals are able to rapidly recover performance after a few trials. However, if the change of the control is outside of this manifold, animals first have a harder time learning this representation, and the performance doesn't get to be as good. They're still able to learn to some extent, but the representation seems to be constrained by some degree to this plane. So in this first part of the talk, I've shown you that latent space models can help us understand neural computation. And these are important because there are recent technological developments that gave us large data sets that we have to make sense out of in order to understand these properties. They hint on the fact that neuroactivity is correlated and guided by a small subset of non-observable latent factors that are the ones driving this computation. With this, I've shown you that we can both understand incoming stimulus, internal processes, which I haven't shown, but here's an example where they make a link to decision choice and or behavioral output. And even more, these latent dynamics are sufficient to control brain-machine or brain-computer interfaces. So I think this is a good time to stop briefly and look at your comments and see if I bored you already or you guys are engaging in the talk. And if you've got some questions, I maybe have time to answer a few. I see a lot of activity in the chat, but I don't think I see any particular question. So I'm gonna keep on going with this second part of the talk, in which I'm gonna talk you about my original research conducted in the Ringberg and Sabin labs. In this project, what we aim to create is a chemical detector. And why so? We've been using animals, and there's a lot of room for application of chemicals detectors. Here is an example where we have a patrol dog detecting explosives or drugs at airports, and they've also been used for a medical application for the detection of tuberculosis here in screening saliva samples from patients. Importantly, we still use these animals because there's no artificial chemical detector that can compete with what animals can do in terms of flexibility and accuracy in this detection. However, although this use seems promising, it comes with a caveat that we have to train animals, and this process is extremely expensive and limited our ability to of odors that the animal can report. Either we make a particular assessment, either saying, in this example, drugs or explosives, or we ask the animals to report both, in which case we wouldn't know which one is present. So our question became, can we build a robust chemical detector from these really sophisticated animal responses or olfactory system, but hijacking just information without having to train animals? In order to do so, we first need some understanding of how odors are represented in the brain. Here, for cartoon illustration, odors are volatile molecules that in mammals, upon inhalation, they enter the nasal cavity and bind receptors in the olfactory epithelium. And here, neurons that express the same kind of receptor project back to the olfactory bulb into a single glomerulus and further down processing in the cortex. If we look at this particular layer of interaction with using calcium imaging, we see that odors evoke rapid and transient spatial responses. For further detail, if we present an odor array, I hope you can see that there's a spatial pattern of neural activity that spans a fraction of the olfactory bulb. So, but if we further characterize this, we can see that it also has a fine temporal structure. If in contrast, we present a different odor, the pattern of activity will be scattered or completely alterated, both in compared to the previous odor, both in space and time. We're indicating that this representation is good enough for us to differentiate between odors. Still, if we present the same odor, here odor A, at a higher concentration, we see that the same representation, I don't have a video, but believe me when I say that the pattern of activity is preserved, but wrapped in the temporal axis, indicating that if we want to also have access to this concentration axis, we need a good temporal resolution for all signals. In order to extract this information, Herrero, a former postdoc in the lab, came up with the following design. He took a mouse, our favorite organism model, and placed a grid electrode on top of the olfactory bulb. And for the proof of principle, we presented odors, a battery of odors at a range of concentrations while monitoring the breathing cycle. Here is an image showing how these electrodes look like on top of the brain. And here are the different sides. And in this particular animal, a given glomerulus was labeled with a fluorescent protein and also expresses channelrhodopsin, which serves as a proof of principle saying that indeed we can capture glomerular activity with this approach. But more informative, can we extract other information, chemical information from this array? And here I'm showing you 3 example traces of a lack of stimulation or air and 2 given odors. As you may predict, now we want to build a detector using these responses. And not surprisingly, we're going to resource to latent space models inspired by the result I showed you before in the locust. And we saw here that once again, for these responses at the level of the glomerular layer in the mouse, different odors expand across different dimensions of this latent space, which could help us decode the different odor identity. Still, here you see that the trajectory evolves over time, so we further characterize at which stages this odor information was present, and we use decoding methods, linear decoding, to answer this question. And here I gave the decoder different windows of this trajectory and try to predict odor identity. And you see that a detector with about 200 milliseconds of these responses is good enough to provide us with maximum accuracy. To further break it down, I provided a detector, a super vector machine with not an increasing not an enlarging window of integration for the decoding, but a sliding window, which once again shows that most of the information is present within the first few 100 milliseconds of the response. This is not surprising, given that animals trained to perform a detection task can report odor identity within 200 milliseconds. Here, in this graph, I'm showing you the proof of principle result of this detection task, where in a 6-level classification, we obtain pretty good classification results for a cohort of animals with over 80% of classification accuracy for the different odors. However, I told you that odors can also vary in the intensity that they are presented at. So we wanted to further investigate if we could extract concentration information from these recordings. And here you see the responses of the electrode sites to odors at a range of concentrations. And just using linear regression, we can see that we're able to predict odor concentration at a wide range of intensities. Good enough, but is 10 to the minus 16 molar a good enough detection threshold? Or better say, in between these 2 orders of magnitude. Since there's no standard in the field of chemical detection, we set ourselves a really high bar, that is animal performance. And in these experiments, D1 in the Bosa lab asked rodents to report chemical detection by training them with associative reward to recognize, in this case, a particular odor at a range of concentrations and only respond when the odor was presented. As we see here, in this task, animals, when they are faced with really low concentrations of a particular odor, they're not able to perform the task and they perform a chance. But as we increase the concentration of this chemical, they're able to decode if the odor was presented or not, becoming almost a trivial task when the concentration was too high. So now we compare our results from the bioelectronic nose or grid electrodes to that of the behavior. And here you see that our ability to detect odor at small concentrations almost perfectly mimics that of the animal. And this was true for the two others we tested. Still, others are not presented alone when in naturalistic conditions. So in the presence of other background odors, can we extract information of our desired target odor? Here again, I'm showing you the performance of our bioelectronic nose in the absence of other masking odors. And as we present or mask with or co-present odors with other masking odors, we see that our ability to detect these odors is impaired, but still we're able to perform somewhat when the concentration is high, and even when the concentration of the masking odors is really high. Once again, this was true for the two others we tested. In the presence of a masking odor, our ability to detect is impaired, but we are still able to detect when the concentration is high enough. So is this all that we can do? Is this the full potential? We have a good system that can identify odor identity and concentration. And because I asked the question, I think that you know that the answer is yes, we can further explode these model organisms and the genetic tools available for those, and to improve the accuracy. In this case, if we know the particular target that we're interested in, which could be explosives, or in this case, PA, and then we know the neurons that respond to this particular odor or chemical, we can overexpress the number of these neurons in the brain, potentially improving our ability to detect these odors. Once again, I'm showing you the average performance of our brain of our bioelectronic nose for all the different conditions. And when we instead record from animals that overexpress these receptors, we see that when expressing the receptor, the neurons, or overexpressing the neurons that are sensitive to PA, our ability to decode this odor was further improved. But not so much was the case for other odors that don't drive the activity of these neurons quite as much. If you're interested in the details and further experiments that we did, stay tuned, because this article is soon to be published. So keep an eye for it. And next, I'm gonna talk to you about something that I'm currently working on that was motivated by a limitation in this current project. So thus far, to perform our classification or detection task, we had to present animals with a battery of odors to characterize the responses, and then only after we train these animals and expose them to those, perform the detection task. So can we use the information that we have from a previous animal to predict in a different animal, and thus bypass the training and exposing animals to a series of or battery of odors? Once again, going back to the biology, here I'm showing you the results from this article where they did in a crazy experiment where they characterized the olfactory tuning properties of a large battery of glomeruli across different animals, and then they measured the distance, the relative distance of those across them. And they showed that across animals, the particular location of these neurons is preserved. So can we use this for the decoding across animals? With my mentor, Christina Sabin, we came up with the following structure. We had, as I said, an animal that we have already exposed to all these others that we've used to decode, and we want to predict other identity in a new animal that we just implanted with these electrodes. So the structure goes as follows. We first generate this low-dimensional space where the odors are represented, and using only information from this source animal, we generate the detection boundaries. And by just projecting the target data onto this manifold, we want to see if we can predict other identity. Importantly, we added some modifications to these algorithms by computing analytically the observation noise terms and generating the latent representation or loading factors from a series of, or a population of source animals. Here I'm showing you, once again, my favorite, these embedded trajectories for 3 different others, and the solid lines represent the source animals, and in dashed lines is the target animals. So here you see that the representations or the trajectories in this subspace follow the others really closely across the different animals. We further use them to predict other labels, and here I'm showing you the accuracy that we obtain with this preliminary approach that I'm still working on. And importantly, we were able to decode with a relatively high accuracy without having to retrain anything for the target animal. And this accuracy is much greater than choice for the both conditions that we tested. However, when we shuffled the representation as a control, our ability to decode was detrimented to the point that it reached choice. So with this, I'd like to summarize. And here I've shown you an algorithm or a technology that enable us to decode identity and concentration from latent dynamics of the olfactory bulb activity, that we can further use tricks to detect both in the presence of a single odor or with backgrounds, and we're currently developing algorithms to further decode across animals. With that, I'd like to thank you all for listening. The NERV group, Zoe, Siobhan, and Jolene for inviting me to give this talk. The NYU Neuroscience community, my 2 mentors, Christina Sabin and Dima Rimber, Eris, who started this program, who started this project, and funding forces, DARPA, and my training grant. Thank you.
Thank you so much, Pedro. So I think we'll get started with the questions now. I see people have some questions here. So let's start. Let's give people a last minute to maybe get some votes in, and then we will start at the top of the list and work our way down. So
Great.
I'm going to
can
hand
everyone
over
just
to
confirm that you
Siobhan
can
now.
hear me? Oh. Sorry, Julianne. I'm just a bit nervous that people can't hear me again. Tonight's not been my night for technology, that's for sure. Yeah.
Okay, it seems like the comments are good. People can hear. Good.
I'm
Okay, so
gonna
I'm
get
handing
going.
over to Siobhan to ask our questions.
Okay, our first question is from Dean. When doing FA or PCA, etc., to find the latent space, is it observed whether the dimensionality, orientation, or any other aspects of the latent space change as the animal initially learns to perform the task?
Really, really good question. This is something, or an area of really interesting active research. There are actually much better methods that evolve or are able to capture these longitudinal changes as another one of these latent factors. And as example is the Gaussian process factor analysis that help us understand also how those processes evolve. So in short, if you were to use factor analysis or PCA that doesn't capture these longitudinal changes, you will see a change in the representation. But more importantly, you can also model it with other methods that capture it and see how it evolves.
Great. Okay. Sorry, I just need to quickly pull up the next question. So our next question is from Chris. What is the computation that these manifolds represent? Is it action or sensing all those experiments, or is it some form of decision making? Can we know this?
Another great question. So another assumption that I try to hide under the rug, but I made explicit through the examples, is that when we neuroscientists are experimenting, we're recording for particular regions when animals are doing a particular task, and we are hoping that those two are related. When looking at these latent representations, we're hoping to find this structure that performs these computations. And by such, we're looking at how either these populations can relate to the task conditions. So in short, briefly, our ability to relate those to actions, sensory stimulus, and decision will be guided by our ability to relate those to the task performance. And by seeing how the trajectories evolve along the different axes, we can see if the particular area is encoding each of those aspects. Does that?
Okay, for our next question, we are inviting our attendee on screen. I've done so, but it seems there's a bit of a delay. So let's give it a second. Otherwise, we'll just ask Siobhan to also ask on her behalf. Okay, tech problems.
Okay, let's jump to Davi's question quickly, and then we can try to get back to Celia. So Davi's question is, why did they do these olfactory experiments on locusts? Are they known for their olfactory abilities?
As far as I know, there's no particular reason. There's the sense of conveniency for using these insects over higher order organisms because it's easier to tackle them in a lab or experimental setting. Additionally, for insects, there's when it comes to olfaction, there's the additional advantage that odors are simply mapped into neural representation without animals having to specifically inhale like we mammals do. So the presentation of odors is really linked to when the odor was triggered. So.
Oh, great. We have Celia online.
Hello, everyone. And hi, Pedro. Good to see you again.
Lovely to see you.
Thank you for the very nice talk. It was very interesting. I think my question is actually two-part. You talked about a lot of different latent variable models, and I was kind of curious as to how you would choose between them because and I think it tied in quite well maybe with the choice of the dimensionality of them. And then which one, if any, would you consider state-of-the-art? So that's like the first part of the question. And the second part, you've been referring to this manifold. How do you what type of manifold? Do you consider it like Euclidean or Riemannian, or have you given that any thoughts? They seem kind of, yeah. What are your thoughts on that?
Thank you for both questions, and they're really good. So depending on which kind of method you use, you're making different assumptions of the underlying properties of the subspace. I try to give a comprehensive and description, and basically when experimentalists deal with it, we start from the simplest one and start increasing the complexity of the models and see which one is able to capture most of our data without making crazy assumptions. When dealing with neural data, factor analysis that assumes independent noise for individual neurons is almost necessary, because there are different populations of neurons that have different firing properties. Where it's also really nice when you incorporate the dynamics, because the properties, as I said, change with the stimulus and the behavior. So the responses also evolve over time. And if you, it'll be nice to also capture those changes in the latent representation. When it comes to the shape of this manifold, is area of really active research. For most studies, we go from a pragmatical interpretation, and we assume that everything is Euclidean and simple, and we go for distances that are easy to parse, and we, for the most part, assume linearity and some of the constraints. So that will be the overall most popular approach to this. But when it comes to state-of-the-art that you also mentioned, there are other methods that are far more complicated that also incorporate non-linearities that the latent space could take in principle any shape or form. And those will be LFATs, which I briefly mentioned, and they are based off autoencoders using recurrent neural networks as a model for the constraining the latent representation. So.
All right. Thank you so much.
Thanks. Lovely to see you.
Yeah, you too. Bye.
Okay. Does that bring us to the end of our questions, Siobhan?
Yes, those are all our questions. Yeah, sorry, David, your last one didn't get any votes, so I'm legally not allowed to answer it.
Okay, so this brings us to the end of our night tonight then. So let me just also share my screen again so we can close off. And there we go. So as always, please give Pedro a virtual round of applause for a very great and very engaging talk. Thank you so much, Pedro. We really appreciate you joining us.
Sure.
And then thank you to all of you in attendance also. We can't do this without you guys, so we are very, very grateful. Then also, please remember to follow our Crowdcast page, and then you will be notified of future events. And look out for our survey at the end of the evening tonight. So we do realize that Crowdcast does have some connectivity issues. So our survey this evening will be specifically focused on sort of sorting those out and seeing what we might need to focus on. So if we could please get some feedback from you, we'd really appreciate that. And then lastly, our sponsors. We'd like to thank Stellenbosch University and Berg, the Biomedical Engineering Research Group. And then we will see you all again in 2 weeks' time on the 20th of May for our next event. Thank you and good night, everyone.
Thank you.